We know that we have our PowerPoint handouts, right? So that has a lot of the core information on it. It is an outline, so there's always a bit more detail than exactly what we see in the slides, but a good chunk of the information that we're going to find on the exam, we can find in those slides, right? Because we said that when we do these lectures, my kind of focus is to help guide you as to the content that's going to be relevant, that's important, and help you understand the concepts that we're going through. So the textbook that you have in whatever fashion, you should be reading that as well, at least using it for the figures and the tables and things like that. But oftentimes they have a lot of content in there. It's hard to kind of figure out what's important, what's not, etc. So that's what we're typically doing in lectures, kind of going through and saying, okay, these are the important things. And as we were discussing earlier, the more time we spend on a particular topic in lecture, the more important it often is for the exam, right? So if we barely touch on it or we don't mention it at all and you're kind of looking through the exam objectives and you're like, seems like we didn't really address that one. It could be that for that semester, it's not really important. I would double check with me if you're going through those exam objectives on your own. Double check and shoot me an email and say, hey, I couldn't find an answer to this in the PowerPoint or in my lecture notes or whatever, just to double check. But you can kind of use that as a gauge as to how important our topics are, as to how much time we spend on them in lecture as well. And then, right, correspondingly, maybe how many questions might be on the exam. So for instance, if we're thinking about the immune system, we had our whole first line of defense, second line of defense, third line of defense, right? And each one, we were looking at cells and molecules that contribute to each. But first line of defense is pretty simple. And we'll go through it as well. But it was pretty simple in terms of, okay, so here's a bunch of different external barriers that are part of our body. And these are different chemicals, and these are different cells that contribute to that. And then after that, we were basically done. Second line of defense got a little bit more complex, where we had more players, and then we had to explain a little bit more how each of those work. So for instance, if we were talking about inflammation or fever, those ones we had to do a little bit of explaining about what it is. What is this process? Because it's more of a process rather than a cell. What is this process? How does it work to protect the body? And what cells or molecules are involved that we can use as an example. So for instance, with fever, a second line of defense, it was mainly a particular molecule called a pyrogen that generates fire. That was the molecule that leads to fever, that temporarily increases our body temperature. So in second line of defense, it took a little bit more explanation. And then third line of defense, when we're talking about our whole T lymphocytes and B lymphocytes and how they get activated, and then once they become activated, what do? We have to develop that clone army, and what are their defenses in terms of getting rid of those specific pathogens. It took a lot more time to go through and understand how those specific lines of defense, as part of our third layer, is working in the body. So likewise, the point there is that, even with those lines of defense that are all part of our immune system chapter, that third line of defense was much more complex. So we spent more time with that. So probably on the lecture exam, you'd probably have more questions dedicated to third line of defense compared to like first line of defense, where it was pretty simple. Okay? It is important for you to be able to also, you always want to be thinking about the big picture as well, right? We want to make sure that we understand our first line, second line, and third line of defense, how this system, this organ system, is doing its job. What are its main jobs for the body in terms of actually supporting us and maintaining homeostasis and things like that? And then what are the different organs? What are the different parts of it? And then we start to get down to the details in terms of how they work and how they contribute, etc. Okay? So don't forget that kind of bigger level of organization too. Okay. So as we were talking about, our main study resources that we have are the PowerPoints. And so we can certainly review those. We can go through those. We also have the exam objectives, which are laying out the major topics in each chapter that we covered that you should be reflecting on and think, oh, Did we do that? I don't know. If we did that, I don't remember. Let me go back to the lecture notes. Let me go back to the PowerPoints and see what we said about that so that I know how, if that came up as a topic, as a multiple choice question or something like that, how I would describe it, how I would define it or something like that. So we have our exam objectives that help us to review the major topics that we are covering, but then also we have particular objectives that are either in bold or in this case they're highlighted in some way. They are kind of focused on because that is going to be our list of potential short answer questions for the lecture exam. So those topics in particular, that means that we could get certainly multiple choice questions on them because like we said, we were covering it as part of our lecture content, but also you could be asked as a short answer question to really describe that in detail in your own words. How does that process work? Or compare these two things in a few different ways so that we can look at their structure and their function and how they contribute to the body. So you have to know those a bit better, okay? So for those exam objectives, not only does it help us to review overall, but it gives us a list of topics that will be your possible short answer questions. So for those, you should be reviewing them all, looking through them at least. You don't have to write them all out. I know that's a lot of time. Not that that wouldn't be a benefit, but it's a lot of time. But you should be looking through them all and reviewing them all. And like I said, at least going back maybe to your lecture notes and saying, okay, for number one, where can I find that answer? And reading it over to refresh that topic in your mind, making your own notes about it, maybe. But for those that are in bold or highlighted, those are the ones definitely you want to focus extra time on. Writing out a complete answer to it, and then once you have that answer, you're like, oh, okay, that's a good solid paragraph about whatever that process is. Then you have to do your time studying that, right? Whether you turn that into a flashcard, break it down into a number of steps and say, oh, okay, this is the process of whatever B cells and how they activate and how they protect the body, you know, and then break it down into those steps and study it, right? Because it's not just, oh, okay, I wrote it down again, that's good. But you need that repetition in there to make sure you're practicing with it so that if you handed that question, that short answer question or those exam objective questions to somebody else and you said, hey, pick any of those ones in bold or highlighted and ask me those, that you would have a good chunk of information to share about it using anatomical terminology, right? So the more general you are, in terms of an answer, if somebody was asking you that question, the more general you are, the shorter the answer that you give. That's a little bit of feedback to yourself, showing you, oh, okay, you know, I know something about it, but I kind of tapped out in terms of my answer after I've said about 2 or 3 sentences. And I know if I have a short answer question, I've got to be detailed enough to answer all parts of that question to describe it accurately, okay? So you want to do those checks with yourself or with somebody else as well. So if you create those flashcards, be using them for practice, but also be handing them to somebody else and say, go ahead and ask me these. Because as I've been, you know, talking to individuals who have emailed me or stopped by my office hours or something like that, you do really want to put yourself in that space of kind of a testing type environment so that, you know, not only are you practicing with the content, but you're practicing in terms of being assessed, being asked questions so that it's not so stressful. So that if you can do more of that yourself, you're like, okay, let's go over flashcards, or let's go over the exam objectives. You ask me and I'll ask you. At the beginning of it, there's a little bit of panic where you're like, oh, I don't know if I've studied these enough. Okay, ask me that one, ask me this one. You get a little bit more feedback in terms of how your preparation is going, right? So ones that you can answer well, great, right? You're getting that confidence. You're like, oh, I'm feeling ready. I like that. And those topics I feel good about, so I can like maybe put those over to the side, and I've demonstrated that I know those. Now I'm going to spend time on those that, oh, maybe these are the flashcards that I didn't do so well on, so I'm going to focus on those. And so I'm going to have to study those more. I'm going to have that person then ask me those questions again, right? So you want to get into that mode of assessing yourself or having somebody else do it so that when you get those questions on the actual exam, then it's like, it's no big deal, right? So I've seen that question before. Here's my answer. Boom, 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 boom. Okay? Okay. And then lastly, remember we have a vocabulary list as well. So that section, the vocabulary section, which is a small section, it's usually like 5 questions or something like that. To a certain extent, that section of the lecture exam should be the easiest section, right? Because here's your list of what's possibly going to be in that section. And we even have, I think I have like flashcards already made up for you. It's just a standard definition, right? One sentence definition. What does this term mean? So even though it takes some practice, it still takes some work. You got to study, you get the different terms. You've got to, get familiar with the definitions. There's no surprises that's going to happen in that vocabulary section, right? You've got your list of possible sections of our answers and questions about what's going to show up there. So make sure that you study that as well. Okay. So in terms of our review, I think we could certainly look at those different resources and just kind of go through them and just review things overall. We could also, at some time, we could go through just questions that you guys have, like particular topics as you're starting to review your content and do your learning this week, things that you've run into that are stumbling blocks where you're like, oh, well, as I'm looking at this again, I'm looking at this figure or I'm looking at this topic, I don't feel exactly right about it. Like I get this far into it and I kind of get lost. So we want to do that this week as well. We want to iron out any details or any topics where you feel unsure about yourself so that we can get that clear in your head so that we can make sure that, again, if you get asked a topic on that topic, a question, that you feel like you have solid footing in terms of knowing how that works or knowing what those things are. Okay? So I guess before we go straight into like reviewing some of our resources, I'll ask you at this point too, do you have anything like right now where you're like, ooh, I'm just waiting for an opportunity to ask about this particular topic, something that you want to review from the get-go? Yeah?
I do as part of the respiratory system.
Yeah.
One of the objectives is to be able to trace an oxygen molecule from the time it enters the body until it crosses the respiratory membrane into the cardiovascular system.
Yep.
So
So that one, do you remember when we wrote all those different steps up here?
Oh, yes.
That's that one.
Okay.
So that is exactly what we want to do. We want to make sure that when we look at those exam objectives, that we have a solid answer in place somewhere, right? So that you've kind of written it out, et cetera. And so that type of thing that I had done last time or the time before, where I'd written out, okay, so we're going to follow this oxygen molecule. We're going to go through the external nares into the nasal cavity, back to the pharynx. We got the 3 sections of the pharynx. We're working our way to the larynx. We're kind of just answering that question. We have it all laid out. And now that we have that clear, of course, that is going to be up to you to be practicing with it so that you could write it out yourself and make sure that you have all of the different steps involved. You're not skipping a step, like you go from pharynx to trachea, like, oops, I forgot larynx, right? So you want to make sure that you're practicing with it as well.
Was that when
you
talked about like the external nares and nasal cavity?
Yeah. Yep. And if you're answering that question, bless you, you can just write it out, right? So you just have to write, the word to, this, to that, to that, to that, or just do a little arrow or however you want to do it, listing it. You could list them in a vertical column or something like that. You don't have to write it in a paragraph format like that, right? So I'm not too worried about a writing class in terms of how editing and formatting and stuff like that. Well, let's take a look at some of those objectives, and that also may help us to jog our memories for things. And then if there are questions that come up at any time during or afterwards, then we can address individual questions too. So if you do have a question, just raise your hand or reach out as we're going through too. I would imagine, right, so this is Tuesday. We're really fortunate that we have Tuesday and Thursday this time. We don't just have one review time, we actually have 2. So between today and Thursday, I do want to highly encourage you to get studying on this if you haven't already, so that on Thursday we can hit those questions where you're like, oh, as I was going through, I got this, I got that, got that. This is a sticking point for me. Can we clarify that? Can we have a little mini tutorial session on that topic so that I have it clear in my head so I can write up my answers or know how that particular process works, okay? Because the more you get into it and see where your help points or your sticking points are, the more we can address those most effectively. So you have a really solid foundation, okay? Okay, Well, let's take a look at some of these. So the first one, we'll take a look at those ones that are highlighted or in bold, okay? So the first one is laying out some of the differences. It's a comparison type of thing. So we had come from our cardiovascular system, and then we're coming into the lymphatic system. And there are some similarities, and there are some differences, right? So what we just want to do is saying, you know, what are these new things that we're learning about lymphatic capillaries and how this system works versus what we already worked on and learned about, and I'm going to change up the word here, cardiovascular capillaries. Because in truth, cardiovascular is a little bit more anatomically correct compared to circulatory, right? So circulatory system and cardiovascular system are the same. Cardiovascular is a little bit more specific. Okay, so in the cardiovascular capillaries and the lymphatic capillaries, there are going to be some comparisons that are going to be the same, right? So they're both capillaries, and our capillaries are our smallest vessels. Of both systems, they're the smallest vessels. So they're both, like we could compare, we'll try to lay this out a little bit. So we can compare their structure. So, and then maybe, see if I can organize this where if we're comparing them, if there's differences, we can list them separately. If there's similarities, we can just say they both have this. So they're both made up of simple squamous epithelium, right? So in terms of their structure, they're both simple squamous epithelium. That's something that they have in common. In terms of differences, we know that these are, and it kind of depends on which way you want to describe it. You can call it blind-ended, right? So these kind of look like little root hairs as opposed to these ones, which are, and this kind of feeds into the circulatory aspect of it. We could say these ones are continuous maybe with other vessels. And again, I'm just throwing this out as a way that I'm phrasing it right now, but if there's a different way that it makes more sense to you in describing it in your own words, that's totally just fine. That's the one of the reasons why we have a short answer question, is so that you can describe it in your own terms, okay? So these ones, right, they are starting looking like little root hairs, whereas these ones are in between our arterioles and our venules, right? So they're continuous, they're in between these other types of vessels as one big circuit. So we see that as well as a difference then. What else do we see in structure? What else did we learn about the structure that was maybe new about lymphatic capillaries?
Slightly larger in diameter.
Yeah, so they are slightly larger in diameter, so a little bit increase in diameter. And remember that we're talking about, and comparatively, we could say, you know, a little bit smaller in diameter if we were doing each part. And remember that we also have other types of lymphatic vessels as well, right? So there are the collecting vessels of the lymphatic system, there are the lymphatic trunks and the lymphatic ducts and stuff like that. But right now we're just focusing down at the capillaries. So what other things? We can take a look back. Yeah, go ahead.
Are we able to compare like their functions too, not just the structure?
Definitely. We can definitely compare their function. At this point, I was just kind of laying that out there. We were kind of focusing on one topic at a time. But functions, for sure, are going to be good ways. And structure and function, anatomy and physiology, those are good comparisons to have. So what were you thinking?
I think the lymphatic, the lymph only goes forward, doesn't go both ways.
Yeah, right. So this is, it has to do with the blind-ended aspect of it. So when we talk about the function, you could get more descriptive. You're saying, right, we're talking about the fluid inside. So this is collecting fluid from the tissues. And if we're talking about function over here in cardiovascular, we could say this is more about exchanging with the tissues, right? Because cardiovascular capillaries, we have things going out of the capillaries and we have things coming back in. So it's a two-way exchange in the cardiovascular capillaries, but in the lymphatic ones, it's just one way. We're drawing it from those tissues and routing it back towards the cardiovascular system. So that's good. In this way, you know, when we talk about collecting and exchanging and stuff like that, in this one we have, for instance, things like nutrients, right, and gases, and fluid for sure. So we have lots of different examples of things that are going across these capillaries in both ways. And in this case, we're only gathering it, so we're only doing one way. So we are doing fluid, we know that, but we're also doing maybe cellular debris. That's not something we usually pick up in our regular cardiovascular capillaries. Pathogens, ooh, right? Bacteria and viruses and things like that. That's something we're not trying to pick up necessarily in our cardiovascular capillaries. So the things that they are picking up are also different, right? In some ways they're the same, fluids, yes, but we're seeing other items that are different that they're exchanging or picking up from the tissues. So that's a good comparison, like what is actually going across these capillary walls between the two. There's a little bit more in terms of the structure as well. So, and again, notice how I'm going from the exam objective, and now I'm headed back to the PowerPoint. I'm going, okay, where can I find an answer to this one, right? So this is where we're starting to see some information about the system overall, about our lymphatic vessels. We use these figures, we're looking at them, we can see, oh, okay, so here's some of that information, blind-ended and things like that. We get to this one, this is a zoomed up view of, our lymphatic capillaries. So we can see some structures, some additional structures there. So that they have filaments, anchoring filaments. Whew. Get a little crazy there. And they have the mini valves in them. And the mini valves in these lymphatic ones allow larger things to be able to enter into these lymphatic capillaries, right? So between the epithelial cells, they can kind of open up and allow some larger substances to enter in. So cardiovascular capillaries don't have mini valves, don't have anchoring filaments. So that's also a comparison you can use in terms of the structure. Okay? So that's a good kind of summary of a lot of different things that we can compare between lymphatic capillaries and cardiovascular capillaries. Now, if we have a question in the bank there as a short answer question where it asks you to complete them, I'm not going to ask you to do like every single comparison you have, right? Usually, if we have a short answer question, it's usually worth 5 points. So oftentimes, I'm asking you to do like 5 different comparisons. And it doesn't matter, you can choose the 5. So if we look here, right? So there's like 1, 2, 3, 4 that we see here when we're looking at anatomy. That's a comparison too, that both of them have walls made up of simple squamous epithelium. So there's 5. One-way system, there's another one. What they collect, there's another one. So there's a number of different comparisons that you can use there, and you would just need to know and relay 5 of them commonly, right? So I try to create those short answer questions where you have options as to what you tell me as well. Now, of course, if we're doing a process, if we're doing blood clotting cascade or something like that, and we have individual steps, well, you're going to have to give me all the steps. We can't just say, hey, name any 3 steps in this process, and it's like all over the place. In some cases, we do have to know all the different parts or pieces, but if possible, I try to give you some options there. Okay. So what else do we have there? Let's see. So that was our first one. Second one is talking about the location of and the anatomy of the main lymphatic trunks and ducts. All right, so let's go back to our PowerPoint for lymphatic. Let's get past our capillaries, which is what we're seeing there. Oh, there's another thing. Look. Right, in terms of what things are picked up, lymphatic capillaries are absorbing and transporting digested fat. That's a unique aspect to lymphatic capillaries, our lacteals, compared to our cardiovascular ones. When we get to the bigger ones, we get to the trunks and then the ducts. So here we see a list of our different specific trunks. And we can see them also coming after that. There's a picture here. We can see them in bold, our major trunks there. And we said that the different trunks that we have drain lymph from different areas of the body. And to a certain extent, their name reflects the area that they're going to be draining from. So all you would need to do is be able to list those specific trunks in order to answer that question, right? You're going to list that jugular trunk and the subclavian trunk and things like that. And then you're going to say, okay, so where are we collecting that fluid from? So the lumbar trunks are going to be collecting lymph from the lower limbs, right? That's the lower back lumbar region. The bronchomediastinal is going to be draining lymph from, in essence, the thorax from the chest, right? Because that's the area that we're talking about there, bronchomediastinal, around the lungs as well as in between the lungs. Subclavian is going to be draining from the upper limbs, right? Just like the subclavian artery and vein that we see here. So it's going to be from the upper limbs. And then from the jugular is going to be handling from the head, right? So just like we have jugular veins that are draining blood from the head, we have jugular trunks that are draining lymph from the head as well and filtering it along the way. So in that way, and then we finally have that single intestinal trunk. So the intestinal trunk is going to be handling a lot of the abdominal pelvic area instead, right? Bringing it in from here. So if you think about it in that way, every part of the body, the lymph from all those tissues, is going to flow into one of those different trunks that we see there. So all you have to do is kind of describe it in that way, about the region of the body that each of those trunks is serving. Okay? So we have the list of the trunks, then like a one-sentence description about the region of the body that they're draining from, whether it's the lower limb, whether it's the upper limb, head and neck, chest, or abdominal pelvic region. That's it. Okay? Now those are for the trunks, and when we get to the ducts, we know that we have only 2 ducts. So these are the largest lymphatic vessels that we have, and those 2 ducts drain. We look at this figure right here, where it shows those 2 major ducts, right? So we see the thoracic duct on the left side, and we see the right lymphatic duct on the right side. So those are going to be draining the area that you see here in color, right? So the green that we see there, that whole area is going to be draining into that right lymphatic duct. And the right lymphatic duct, both of these ducts, drain into and return the lymph into the cardiovascular system at the union of the subclavian vein and the internal jugular vein. Both of them, that's where they hook back in, okay? So right lymphatic duct does that on the right side, and you can see right lymphatic duct is going to drain lymph from the right upper limb, right upper torso in terms of right thorax, and right side of the head and neck. Everything else that you see there in pink is going into the thoracic duct, which is over on the left-hand side instead. So the similar regions of the head and neck and the chest and the upper limb, but on the left side, plus the abdominal pelvic region and the both sides of the lower limb. All of that goes into the thoracic duct. Okay? So for that, you could describe that if we had a short answer question, because we see this question's in bold, but you could also have like multiple choice questions where it's like, oh, I've got lymph that's being drained from the right wrist or something like that. Where is that going to go into in terms of a trunk? So trunk we just went through, so that would be going into a subclavian, be going into a right subclavian trunk, right? And it maybe would list it as a multiple choice question. Is it the jugular? Is it the bronchomediastinal? Is it the intestinal? And we said subclavian handles the upper limb. And then also you could have a follow-up or a paired question where it's like, okay, yeah. So we said it's on the right side, the right wrist where it's coming from. What about the duct? Is it going into the thoracic duct or is it going into the right lymphatic duct? Well, it's on the right side and it's the right upper limb, so it's going to go into the right lymphatic duct. Okay? So just thinking about how we would assess the information that we were just talking about, right? Whether it's a short answer question, whether it's a multiple choice question, etc., right? Okay. So that was for our second one, that one that we see there, in terms of talking about the trunks and the ducts and the areas of the body that they're handling. So those are the two that we have for lymphatic system. While we're looking at this right now, and you're looking at those other objectives, do you have questions about some of those other objectives? Anything that seems totally unfamiliar? While you're looking at that, also say that oftentimes when you're going through these objectives, and some of us have been going through these objectives in office hours, etc., you will find some overlap sometimes between these questions, right? Because whether it's the structure and then the function, we know that those are always going to work together. Or if we're talking about like a bigger level question and then a smaller level question, well, the smaller level question will feed into the bigger level question too. So for instance, on number 2 right there, it says, describe the anatomical adaptations which enable lymphatic capillaries to absorb tissue fluid. All right, so we had looked at this, and we looked at some of the anatomy, right? And we did mention that they have mini valves, which will be these separations between the epithelial cells, which will open up and allow bigger things like cellular debris and cells to come into the capillaries, the lymphatic capillaries. We wouldn't get that necessarily in our cardiovascular capillaries, right? So this is the anatomical adaptation, having those mini valves, which allows these lymphatic capillaries to pick up more large level items and return them to the cardiovascular system. So you will see that overlap where each question you're like, oh, I kind of answered that in this question over here. And that's okay, right? It's a good way of kind of, again, getting that repetition and thinking about these things as well. Other things that as we've been looking through these, so for the lymph node, explaining the gross anatomy of the lymph node, that kind of ties into a bit of what we're seeing in here, because we have a cortex and a medulla for that. And then this is just looking at a little bit more of the details of the vessels going into the lymph node, coming out of the lymph node. For these ones, I wouldn't worry too much about the cortical sinuses and the medullary cords specifically. We just know that in the lymph nodes, their main role is to filter the lymph, right? So it's a filtering point where we're going to be exposed to pathogens or cellular debris, and we're going to try to take them out before we dump this material back into the cardiovascular system. So we have a number of lymphocytes, B lymphocytes, T lymphocytes, macrophages, and things like that are hanging out both in the cortex, in those follicles, as well as in the medulla. They're cords, they're connective tissue cords, they're kind of hanging on there like ropes or vines and just waiting for the fluid to come by and checking these things out. So not a lot of critical detail having to do with the sinuses and the cords, but we do need to know that the lymph nodes themselves are different types of lymphocytes and macrophages and things like that in it, because that's what's doing the filtering. That's what's removing these pathogens, etc., from the lymph that we've collected. Okay. Questions on lymphatic before I move into immune? All right, so it looks like these other questions are just asking a bit more about lymphatic tissue and lymphatic organs that contribute to the system. So we went through and we said, oh yeah, so the tonsils are part of it, and the spleen is part of it, and the Peyer's patches and stuff like that. So you'd want to be able to describe each one of those organs and kind of what they do, where they are, and to a certain extent, how they're protecting us. Now, most of these have resident populations of the same types of cells inside of them, and that's how they're doing their job of protecting us. But describing where they are and their general function is going to be important. Okay, so now we get to immune system. And with the immune system we have, right, we spent more time with it for sure. Lymphatic, we did 1 lecture with it. Immune system, we did like at least 3 lectures with it. Now, I'm not saying there's going to be 3 times as much immune questions versus lymphatic, but there's definitely going to be more, right, because we spend that much more time with it. So, likewise, in terms of your studying, you want to make sure that you're going to spend more time with the immune system because that's a lot of detail in there, right? Okay, so the very first one is talking about the difference between our non-specific and our specific immune responses. So that one, and sometimes when we have when we have essay questions, sometimes it'll bring some of these exam objectives together. So for instance, with something like that, we're looking at a big picture question where we're like, okay, we talked about our defenses and we laid it out as first line of defense, second line of defense, third line of defense, right? So when we're doing that, we want to make sure we understand, well, even with that, what does that mean? Why do we have first, second, third line of defense? So when we were talking about that, we use some terms to differentiate or compare each of these lines of defense, right? So the first line of defense, and for each of these, we're saying whether they are specific or non-specific, and we're saying whether they're external, internal, okay? So we're just using those words, but you would want to be able to describe that, right? That external defenses or whatever, barriers that are keeping pathogens from getting into the body. Internal defenses, the pathogen, the bacteria or virus or whatever, has already entered the body, and now we're trying to attack them or get rid of them from within the blood or within the tissues or something like that, right? So just be able to describe it as well. But when we're talking about these three lines of defense, first line of defense, is that internal or external? External, right? So it's external. So these are our walls and things like that that are keeping things from getting inside. That means that after we get past that, second line of defense is going to be internal. Third line of defense is also internal, right? They've already made it inside, and so now we're having to battle them within our own tissues. So first line of defense is external. Is it specific or non-specific? Non-specific. And second line of defense, now we're inside. Is that specific or non-specific? That's non-specific as well. And then finally, our third line of defense is internal, and this is now specific. Now, remember that non-specific is also known as innate. So this is one of those cases where you can choose to call it innate or non-specific. Those are both acceptable. They're both correct. But you want to know what each one of them means, and you want to know that in this case, we're referring to our first line of defense as external and non-specific or innate. So like if you get a question and it's like, oh, okay, so what is the, which line of defense is external and innate? You got to be thinking about, oh, I don't know innate. I studied it as non-specific and specific. So if there's an alternate term for something, you've got to, it's okay to use the one of your preference, but you got to at least recognize the other one. So if you see a question or something like that, or a topic, you know what we're talking about. So innate means that you're born with it, okay? As opposed to later on, you have the adaptive defenses that change, doesn't go there, it goes here. The adaptive defenses that change through time. So in these ones, in the innate ones, you basically have all of these different examples that we're talking about as part of first line of defense and second line of defense. Really, you have them from the day you're born. You don't have to develop them through time. But third line of defense, you do have to develop one. That one gets better through time. It changes through time. So we call it adaptive, okay? Physiologically adapting. And then between specific and non-specific, we have to know what that means as well. It's great to have these little words. This is the basic information you want to know. Is it internal or external? Is it non-specific or specific? That's good for a very basic answer, but unless you can describe what that means as well, you're not quite there yet, right? So we talked about innate, and we talked about adaptive. Non-specific and specific is really getting down to talking about those antigens, right? So those are key terms, really key terms for the immune system. The antigens, which are those molecules that are attached to cells that help to identify them. And we know that we have self-antigens and we have foreign antigens. The self-antigens identify our own cells, molecules stuck to our own cells that identify them as part of us. Foreign bodies and pathogens, etc., have foreign antigens, their own little ID molecules stuck to the surface of them that help us to identify them as something that doesn't belong in us, okay? So when we're talking about non-specific and specific, the non-specific defenses are just getting to the level of, okay, are you part of us? Are you on my team or on the opposite team? Okay? So for instance, if you're watching sports, if you're watching football, the two different teams have different uniforms, right? And usually those uniforms are very different in terms of their color, right? It would not be helpful if they had the exact same color of uniform and the exact same pattern. Because then when you're on the field, how are you going to know who to protect and who to attack, right? So they make those uniforms very distinct. So even in the heat of the moment, you're just looking at my color is green, that color is red, anybody in red I'm attacking. Very easy to do, right? So that is kind of like non-specific. We're looking for foreign antigens versus self-antigens. Anything foreign, we're going to attack and try to get rid of it, okay? So how do we contrast that with specific? So specific means, yeah, we start with that, but we need to know exactly what that pathogen is because we have a very, very specific cell that's going to be able to deal with it. So if we take that back to the football analogy, it's not just like, oh, my team is green, that team's color is red. I'm supposed to cover a very specific person, right? So if I'm supposed to cover that wide receiver, I need to know maybe what his face looks like. I need to read in more detail his name and his number, right? Because then I can tell, oh, that is the specific one, and I'm the specific cell or person that's going to deal with that, right? So that's the level of specificity that we need for third line of defense. So we care, not only is it self and foreign, and yes, that's a foreign agent, but okay, but what is it? Is it a virus? Is it a bacterium? And what specific species is it? What specific one? By looking at that antigen on its cell surface, we're going to find we need a particular cell with a complementary receptor on it that can identify that particular one and deal with it, right? So not only does it depend on whether it's a virus or a bacterium or a fungus or something like that, but we care about exactly which type of virus it is, which type of bacterium it is, because we've got to figure out what cell is going to deal with that, what T cell is going to deal with that. Okay? Okay. So in this case, that first exam objective is really just kind of making us aware of, okay, what's the organization of our defenses and what does it mean in terms of, like we said, internal, external, innate, adaptive, etc. Okay. So from there, we get to, you can see how the highlighted ones are really focused on particular examples within those first line of defense, second line of defense, third line of defense. And so we can see like number 3 right there is listing a bunch of different structures, or in this case chemicals, molecules, that are part of either our first line of defense or a second line of defense. Because we said that with each of these, we need to be able to list cells and molecules that contribute to them and then what they do. So for instance, gastric juice, that's the first line of defense. Lysozymes, that's the first line of defense. Sebum, that's the first line of defense. Interferons, complement, and transferrin are actually second lines of defense. And transferrin, interestingly enough, sometimes I forget to mention that during lecture, but transferrin, as the name indicates, ferrin, ferrous, Fe, if you look at the whole periodic table up there, refers to iron. Iron is something that bacteria use to replicate. So sometimes when you are ill, what your body will do is it will use these molecules called transferrins, which will take iron out of availability in your body, and it will sequester it. It will hide it kind of in the liver so that your bacteria don't have access to it, which means that they can't reproduce as well, which means they can't spread as readily, which means they're kind of contained in terms of their population, which makes it easier for us to get rid of. But the point of this one is it's having you review specific examples of first and second line of defense. So not only categorizing where they go, but then also describing kind of what they do, right? So for that one, if we were looking at, okay, where am I going to find some answers for this in terms of describing what those do? we would go back to the immune system lecture. There's our first and second and third line of defense and stuff like that. And this figure right here does a good job of helping you to see some of those different examples of first and second and third line of defense as well. So at the beginning, it says surface barriers is first line of defense. And as we go through the slides afterwards, now we're seeing a list of these external defenses, cells and molecules that we would use as examples of first line of defense. So the cells of the skin, right, the stratified squamous epithelium, that would be a good example of a cell as part of first line of defense. Just physically being there as a barrier to prevent things from getting in, okay? The keratin, though, inside of it is a molecule that helps to create strength, that strengthens those epithelial cells so that they don't just flake off as readily, et cetera, and they can be resistant to certain types of chemicals, right? So the keratin is an example of a molecule that we have in those cells. When we talk about the mucus linings, whether it's along the respiratory path or the digestive path or the urinary path or something like that, we know about those from 201. So we talked about, okay, so those are going to be mucosal cells. They secrete mucus, right? So the epithelial cells, whatever it is, simple columnar epithelium or pseudostratified ciliate columnar epithelium, whatever, those epithelial cells and the goblet cells within them, those cells help to create a barrier, much like the skin, the stratified squamous epithelium. Those that are lining the mucus and hollow pathways along the body, those are cells that can contribute, and then they produce, like the goblet cells, produce mucus. And so mucus itself would be a molecule as a first line of defense as well. It's like flypaper, right? It catches pathogens and dirt and debris and things like that, and then can be moved out collectively. What else do we have? So there's talking about epithelium again. We talked about sebum. So sebum is, in essence, coming from our sebaceous glands. And so that's some of the oil that goes onto the surface of the body that is acting as an antibiotic. So it's got chemicals in it that have antibiotic properties. We have another chemical here. So the mucosal lining of the stomach secretes hydrochloric acid. Hydrochloric acid denatures proteins, basically chemically digests, if you will, bacteria that you may have eaten, etc. So that is another molecule as a first line of defense. There's some of the lysozymes that we talked about. So lysozymes are enzymes that we find in certain secretions. So saliva and lacrimal fluid. So those lysozymes are going to work to kind of degrade bacteria or chemically degrade, and if you will, digest those different types of pathogens. And then we already talked about mucus as well. So there's a few more chemicals that we see there as part of our first line of defense, right? These secretions, these layers that we are putting out on the surfaces of the body, kind of lining the inside or the outside of the body, that are going to protect us, okay? So all that's first line of defense. So you don't have to know every single one if you had an essay question, but you need to be able to list enough of them, right, to answer a 5-point question. You would also, on the flip side of it, you would need to be able to recognize it. So for instance, a simple multiple choice question might say, sebum, is that part of first, second, or third line of defense, right? So that way, as you're learning, you're going, okay, first line of defense, I listed all these things. What if it's like doing your flashcards the opposite way? So what if I have first line of defense and I have to be able to notify or kind of recall those things on the back? What if I look at all the things on the back and I have to recognize, oh, okay, I'm looking at first line of defense as opposed to second line of defense as opposed to third line of defense, right? So using those flashcards in both directions can be helpful as well. Can you recognize the cell or the molecule by just reading the function of it in terms of what it does? And that way, if it came up like that in a multiple choice question, it could be helpful. Okay, what else? Oh, so we got into second line of defense. So we had complement, we had interferons. We talked a little bit about transferins there. So we get into second line of defense right here. And now we have different examples of cells and molecules as part of second line of defense. So from there, we see, okay, so there's some cells, phagocytes, those are cells for sure, right? Phagocells are phagocytic, they eat up our foreign agents after they've identified them as foreign, right? And actually, coming up, we even have a slide where it talks about and shows you how a phagocyte does that, where we see our pseudopods reaching out and grabbing onto it, latching onto it, bringing it into the cell and combining it with the phagosome, the little bag that it's in, combining it with the lysosome, those enzymes to digest it, and then kind of, if you will, barfing out through exocytosis all of those little pieces. Now, interesting part about this that we learned later on is that a number of our phagocytes will act as antigen-presenting cells, right? So rather than barfing out all of these different components of that foreign agent, they will hold on to some of those foreign antigens and intentionally put them on their cell surface and display them as kind of like a wanted poster and saying, hey, we're infected with this as well. So this is where kind of our phagocyte straddles the line between second line of defense and first line of defense. They'll eat a variety of different things. They don't care. So that's why they're non-specific. But they do aid our third line of defense and say, hey, I did eat this one specific thing, and help to kind of stimulate an immune response on the third line of defense in terms of T cells or B cells interacting. More the helper T cells in that case, but regardless. All right, so we have phagocytes. We listed some examples of phagocytes. There's the natural killer cells as well. So the natural killer cells, as we mentioned, they are a cousin of our T lymphocytes. And in fact, after we went through the third line of defense, now that we look at this, we see, oh, they release perforins. The perforins, the weapons that our natural killer cells have, are some of the same weapons that our cytotoxic T cells have. They just don't care who they are using them on. They will basically, as long as it's a foreign cell, they'll use those weapons on any of them, right? Whereas the T cells and the B cells, they have to match up with a very particular pathogen instead. Okay? So that's second line of defense as well. There's our inflammation. That's more of a process in terms of inflammation, right? But there is a chemical there, histamine, which allows the blood vessels to get leakier, allows these defenses to get to the site of infection. That is a chemical as part of second line of defense. Mast cells produce the histamine, and so that's a cell of second line of defense. Inflammation as a process, as we were talking about, you do need to understand it. Understand that this is where our fluids as well as our cells that are within the bloodstream are able to migrate to the area of infection. And so make sure that you refamiliarize yourself as to what margination is, what diapedesis and things like that, right? So margination is where they're kind of slowing down and sticking to the blood vessel wall. And diapedesis is where they're actually squeezing out and then moving towards that site of infection. So diapedesis is that movement instead. And then what else do we have there? Oh, that's where we're getting to things like interferons. So interferons was listed in that exam objective. So we need to know, have a definition, be able to write down, for instance, on a flashcard, okay, interferons in the front of the flashcard. Maybe in the core of the flashcard, second line of defense, right? Because you want to organize every time we're talking about a cell or molecule, where does it go in terms of our lines of defense? And is this a cell or a molecule? Is that a cell or a molecule? That's a good thing to know as well. Each time we're listing a defense, is it a cell or is it a molecule? This is a molecule. So interferon is a molecule that is released by cells that are already infected with the virus, okay? So it's released by a cell that's already diseased. It's going to end up dying, but it releases interferons that then send an alert to the cells in its neighborhood, within its tissue, telling them to basically lock their doors, right? To secrete these virus-blocking enzymes and seal themselves off so that they will not become infected like this original cell was. So it's an alert molecule that is, in this case, particular to viruses, but it helps to keep that spread of a virus happening, okay? So interferons have to do with alerting other cells that a virus is in the area and keeping that virus from spreading to those other cells. So then we had interferons, and then we had the complement system, or the complement proteins, right? So these that we see here inserted into the plasma membrane, those are complement proteins. So complement proteins, what are they doing? They form a membrane attack complex. So they collectively work together to form these big holes in the plasma membrane of a foreign cell. And when we create a big hole in the plasma membrane of a foreign cell, then that's going to cause it to lyse, right? It's going to basically poke a big hole in it, and it's going to kill it off. So that's what complement proteins do. Now, complement proteins as second line of defense, it doesn't matter, again, exactly what virus of what bacterium, let's say, it's going to be working against. It just knows that it's foreign, and so it's going to assemble and make a big hole in it in order to kill it off. Now, interestingly, down the bottom of the slide, it also talked about how this is where we see an overlap between second line of defense and third line of defense, like the phagocytes that provide some sort of defense, but then also alert our third line of defense or specific defenses. These also can work in a similar way, where once complement proteins are inserted into a foreign cell, they can serve, number one, as a chemotaxin. So remember, chemotaxis is where our different white blood cells are kind of smelling chemicals and moving towards that site of infection, right? So when there's complement proteins in a particular area and they're inserting into those foreign cells, the presence of those complement proteins act as a chemotaxin. It kind of puts, if you will, a smell, right, an infectious smell into that area that will draw more of our white blood cells, more of our lymphocytes and things like that. So it can act as chemotaxins, where our other defenses, more like phagocytes and things like that, can show up. And as an opsonin is where we have molecules, typically proteins of our own, that are now stuck into this foreign cell, and they make it easier for a phagocyte to identify it, because they see all these different things sticking off, and to grab onto it, right? Because we saw those pseudopods reach out and grab that foreign agent to bring it in. And if we have these different flags, these different molecular flags sticking out, then it'll help it to grab onto those and then bring it in. So complement proteins sticking out, antibodies sticking out of a foreign agent also can work in this similar way. Okay? So any of our defensive proteins that are stuck to a foreign agent can help to draw in other defenses to get rid of it. Draw them in and also help them to grab on. Okay. What else do we have? So those were all aspects of either first line of defense, second line of defense. And in fact, when we got to 5 here, now we've kind of wrapped into it, and you can see how we've answered some of 5 as well, right? So we were going through second lines of defense, and we said, oh, okay, so let's see. Inflammation was a process of second line of defense, and now we see, look, there's diapedesis that we see right there. Chemotaxis, moving to the site, being drawn by chemicals, that's right there. When we talked about the complement proteins as second line of defense, we mentioned opsonization, where it makes it easier for those phagocytes to grab onto it. We mentioned a bit of phagocytosis, and we mentioned how when they're leaving the blood vessels, they have to do that margination as well, right? Slow down at the site of infection. The only thing we didn't talk about as we were going through inflammation is pyrogens. So pyrogens has to do more with fever, and it can be associated with inflammation to be sure. But our pyrogens, like we see here, so now we see in our PowerPoint again, pyrogens are going to be the chemicals that are released that generate a fever. So remember, pyro means fire, so that's generating an internal fire. These chemicals that are either released by a variety of different white blood cells or even by infected tissues will then cause that temporary increase in temperature. And that increase in temperature is not burning our pathogens out, right? It's not sterilizing it. It's not an autoclave. It doesn't get that hot. What it's doing is it works to increase our metabolic rate, which then causes our defensive cells and our repairing cells to move faster. It causes our responses to move faster. So it's kind of like if you're in the emergency room or something like that, and they're like, oh my gosh, we've got a bunch of people coming in, they're infectious, and so you've got to suit up and we got to move now because we know that this infection can spread, and so we need all you guys who are just coming off a break and you're feeling, oh, I'm not up to it, you need to get together now because this is going to be a serious situation. So with fever, it increases all those different cells that we're talking about, our phagocytes and our T cells and stuff like that. Their movements and their patrols and their actions happen faster instead, okay? So that's kind of the role of fever overall, as well as, remember when we were talking about transferins and things like that? That's also involved there. So it encourages that movement of some of those nutrients, if you will, away from the microorganisms, from the bacteria, et cetera. Okay, so that was taking us in through our second line of defense. So that was all second line of defense there. And now we're starting to get into third line of defense. And we can see how third line of defense, there's as many questions in terms of short answer questions having to do with third line of defense as there are combining first and second overall, right? Okay, so before I address any of those, you have questions? Yeah?
Can we
Gio.
go on number 5, opsonization?
So opsonization, we had mentioned that opsonins or opsonization is when we have molecules like complement or antibodies that are attached to foreign cells. And those molecules that are attached to the foreign cell make it easier for a phagocyte to grab onto it, to ingest it. So opsonization is when we're marking foreign cells and making it easier for phagocytes to eat them.
Thank you.
Mm-hmm. Okay. So when we get the third line of defense, when we're getting to T cells and B cells, we have a natural separation in what we learned in terms of, okay, what do B cells do and what do T cells do? And each one of those is a fairly big topic, okay? But before we get to that, we have to understand how our immune system or how our third line of defense is going to identify these pathogens specifically. And that comes down to our MHC molecules. So that by itself is kind of a bigger topic. So we have MHC. So first thing is, what does MHC stand for? What does it stand for? Major? Yeah, histocompatibility complex. So that's our MHC, and we know that we have two types. And really what this is we're talking about an MHC molecule. That's one of our own self-antigens, okay? It's one of our own self-antigens that is made into a complex because it's a self-antigen combined with a foreign antigen, okay? For the most part, if we're talking about like infected cells and things like that. So we've got two different types. So we've got type 1, so we can call that MHC1. And then we have MHC2. Okay. So let's do some comparisons. Why? We have two different types. They've got to be different in some way. Probably structure, probably function, etc. So we need to know some details about them. So what can we say about them? We can say the cells that they're found upon. So like what type of cells have, these are all part of us, right? We said that these are self-antigens. Does that mean every cell in our body has both MHC1 and MHC2? No, not exactly. So MHC1 cells are found, our molecules are found on almost all cells. Almost all body cells, put it that way. Versus MHC2 molecules are only found on what, do you know?
Certain cells in the immune response.
Yep, definitely certain cells in immune response. So some of our defensive cells, there's a particular type of defensive cell that's going to be showing this MHC2 molecule. It's basically going to be saying, hey, I ate this. Can everybody help me with it? And showing it off. What does that sound like? So we saw when a phagocyte is eating a foreign agent, we said it's getting rid of a lot of the cellular debris from that foreign agent, but it's holding on to those things, those molecules that identify that foreign agent. Those are foreign antigens, right? The tags that were on the surface of it, the face of it, if you will, it's holding on to it because that's what we're going to be able to recognize. So it is presenting those foreign antigens to other agents of our immune system. So MHC2 molecules are on antigen-presenting cells. Those are the cells that have MHC2 molecules, and only those cells. Antigen-presenting cells. Now what we saw when we were looking through the notes is that those sometimes are abbreviated APC. You would need to write it out at least the first time, and then afterwards you could abbreviate it APC. What are some examples of antigen-presenting cells? Because there's a few different ones that actually do this type of, hey, I ate this. Generically, what type of cell were we already talking about that's an eating cell? Yeah, right? So phagocytes, general phagocytes, are going to be our main antigen-presenting cells. And of the phagocytes, we have like macrophages. Those are like our monocytes that are now on patrol, our big eating cells. Our dendritic cells that look all spiky, like they have dendrites on them. And even you can get some B cells and eosinophils acting as antigen-presenting cells. So those are examples of phagocytes, and it's only the phagocytes that are the ones that are presenting their antigens. Only phagocytes that are going to have those MHC2 molecules. So they'll have MHC1 as well, but they'll have MHC2 having to do with their specific function in terms of antigen presentation. Like I ate this and I'm showing it off. Okay? Okay. So almost all of our cells have MHC1 molecules. Okay, now let's talk about what else about them. Let's look at our PowerPoint. How about that? That can always be good. That's what we're trying to do, is kind of reflecting back and using the PowerPoint to help us answer things. Oh, look. So there it is. So slide 25 addressed MHC proteins, MHC molecules, right? There's a definition right there. And here's where it says class 1 and class 2 between what cells. Now, we have provided a better answer than just saying certain cells of the immune response. Now, that was a good first step, but then we narrowed it down to, oh, okay, phagocytes. And we said, here are some examples of phagocytes. So that's better still. From there, I would say, if we were talking about answering more information about these, I would go to, they're mainly used associated with the T cells, interestingly enough. So like this one here is our first image that does a good job of showing MHC1 and MHC2 molecules. We see it there, and we see it here, like that. So that slide, what slide is that? That's slide 41. That's a good figure there. And then slide 46 and slide 48 have good images that are also showing us our MHC1 and MHC2 molecules. Okay. So let's see, how are we going to phrase this? So for MHC1 and MHC2, how do I want to phrase that? I'm trying to figure out a way to say, what are going to be the cells that use or are recognizing MHC1 and MHC2 molecules? I'm trying to figure out a way to say that a little bit more concisely. But what do you got? You got any suggestions? Hive mind type of thing. I don't know. But you know what I'm trying to say? I'm trying to talk about what specific immune cells use this molecule. Find with it, read it, use it, I don't know, something like that, you know? Because that's what these MHC molecules are for, okay? All right, so what we can see up on the board there is an example of a cytotoxic T cell, and we're seeing the activation of a cytotoxic T cell, and what you can see right up there is an MHC1 molecule, right? So on this slide, we can see how an MHC1 molecule, which is this thing that we see right there, how we see a cytotoxic T cell interacting with an MHC1 molecule. All right, so what are we seeing here? We're seeing an infected body cell displaying an MHC1 molecule. As we said, MHC1 molecules are on almost all body cells. Now, if we were talking about a healthy body cell, that healthy body cell would have this green part of the MHC molecule, and it would just only have that green part and not that foreign antigen that we see attached to it. That little part that we see right there, that little kind of purple sphere, that's actually a foreign antigen, right? So that is a foreign antigen. I'm not going to be able to write that small enough, but you see, ooh, look. That's a foreign antigen right there. This is a foreign antigen. And so if we were talking about a healthy body cell, it would have the green MHC1 molecule that says, hey, I'm on your team, but it wouldn't have any foreign antigen associated with it. So then a cytotoxic T cell coming up to it would look at it and it would say, oh, okay, you're on my team. It looks like you're not infected. I don't see that there's any bad stuff oozing out of you, etc. So I'm just going to pass on by, okay? So if we're talking about a healthy cell, that MHC1 molecule is just used in order to identify that as a cell as part of us, right? So, but if it's an infected cell, then it's going to be our cytotoxic T cell, which is going to be the one that is reading and looking at that MHC1 molecule and say, hey, are you infected? No, you're okay. Oh, no, wait. Look, there's something oozing out of you. There's a part of a foreign antigen that you have displayed on your surface showing me that you're sick, that you need to be killed off, that you need to be gotten rid of, okay? So cytotoxic T cells are going to be using our MHC1 molecules. As they're going through and doing their immunosurveillance and they are looking at all of our cells to see if they're healthy or not, those are the ones that are going to be read, the MHC1 molecules are going to be read by our cytotoxic T cells, okay? And if that MHC1 molecule has a foreign antigen as part of it, that's going to set off a signal to kill off that cell. Now in this case, what we're seeing here is, look, there's our MHC2 molecule, okay? So we see here an antigen presenting cell, because we said an antigen presenting cell is the cell that's going to have an MHC2 molecule. And we see that the MHC2 molecule is going to be the one that antigen presenting cell is intentionally showing off whatever it ate, showing off that, again, foreign antigen, like that. And what is it showing it off to? It is showing it off to, instead, a helper T cell. So what we're seeing here is that it's a helper T cell, or helper T cell individual one, that is reading an MHC2 molecule. Okay? So in both cases, we basically have some sort of cell that is showing off to a T cell. It's showing off an antigen that is part self and maybe part foreign, right? So in terms of an infected body cell like we see back here, it was showing it off and saying, oh, this is inside of me, please kill me. And in this case, with the antigen presenting cell, it is saying, no, 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 I took care of it. I'm not infected. I ate it and I destroyed it. But I'm just showing you the pieces of it so that you can potentially help out in case there are more of these things around. Okay? And that's what we see going on right here, right? So if it has that foreign antigen with it, then that's going to activate, in this case, our helper T cell. And what we're going to see there is we're going to see proliferation, right? So that step, whether it's a helper T cell or whether it is a cytotoxic T cell, proliferation is where we are creating this big clone army that then we can send out in the body, because that's the particular cell that has the right shaped receptor for that particular foreign agent, foreign antigen that's been shown to it, okay? So, in a way, right, even though we were looking at that exam objective and we were looking at it as just talking about MHC1 and MHC2 molecules like that, it also helps us to kind of get some of the answers for these other ones as well, right? So we talked about a little bit about foreign antigens versus these MHC self-antigens. We talked about antigen presentation, both in terms of an antigen-presenting molecule, saying I ate this, but also if we have a sick molecule going, oh, look at this, it's inside of me, please kill me. In both cases, that's antigen presentation, right? And then we also then tied it to different types of T cells, cytotoxic T cells and helper T cells, okay? Now, what we haven't gotten to yet is talking about, number one, okay, what are cytotoxic and helper T cells going to do? Because every time we say, oh, this is a cell, this is a molecule, we have to say, what does it do? How does it help us? How does it protect us? So we still have to do that. And we haven't quite yet talked about B cells. We did mention B cells in terms of they can be an antigen-presenting cell, but we haven't talked about their role yet in terms of that humoral or antibody-mediated immunity. Okay? So we'll talk about that next time as well. So we'll go over that. We'll finish up going through immune system. We'll go over respiratory system and those 4 different functions, etc. But then also bring your questions, bring your own questions and stumbling places so we can iron that out for you too.
Two.